Hi, everybody. I'd love to introduce Jennifer Pett-Ridge from Livermore National Lab, and she's been doing a ton of work on measuring, modeling soils and the carbon storage, and just recently started a soil ah a soil energy initiative, like an Earthshard initiative, to model it and improve our carbon sequestration. Welcome, Jennifer.
Thanks so much, Greg. It's great to be with you all. And I am going to cover kind of a lot of ground today, but the goal is to tell you a little bit about the work we're doing in what we call farming carbon. And so this is going to be a mixture of modeling of what's going on at the national and even global scale, but getting down to even some of the tiniest microorganisms and viruses and modeling their effects on the soil system, what we call biogeochemistry, kind of how the whole package of soils work together. I am a senior staff scientist at Lawrence Livermore National Lab. I also have adjunct positions at UC Merced, and I sit at the Innovative Genomics Institute at UC Berkeley. So I sit in a lot of places. I actually live in Berkeley, though, so that's where I am today. And I always like to start off by giving credit to those who really deserve it, and these are the wonderful postdocs, collaborators, staff scientists, even some technical staff who have really done the majority of the work that I'm going to talk to you about today. So I want to give them credit from the get-go. This work is almost all funded by the Department of Energy, both really fundamental work that we do in a project we call Microbes Persist, and then much more applied synthesis work that we've done as part of a new analysis, the roads to removal analysis of carbon dioxide removal in the United States. And I believe that you fairly recently heard from Corinne Scown, who was part of that team. So I'm hoping that you're somewhat familiar with that effort and that report. So let me just set the stage by talking to you a little bit about what's going on with soil carbon on our planet. Over the last 150 years, we've lost close to 500 gigatons, and a gigaton is 1 billion tons of CO2 equivalents. And that's largely just due to the disruption of agriculture. We're practicing agriculture on almost half of the land surface of the planet. So every time we plow, every time we harvest, we are causing losses, largely because we're literally putting carbon in front of microbes and allowing them to degrade it. It's simply through the rearrangement of the soil system. There's also a certain amount of carbon that's lost simply because of erosion, and that carbon is not necessarily going to be going up into the atmosphere, but it's being redistributed across the surface of the planet. Now, you're maybe thinking right now, okay, why do I care? Well, soil carbon is part of what we think of as soil organic matter, is incredibly important to healthy soil systems. It holds water, it holds nutrients, it holds a diversity of microorganisms, which can both cause and protect from plant diseases, all sorts of things. It's a really critical foundation for healthy human systems, healthy agriculture, all kinds of secure systems that we really need to rely on. So the question is, can we put it back? Can we take some of the carbon out of our atmosphere and get it back in the ground? Well, if you read the literature, there's a whole bunch of papers here that will tell you absolutely we can remove gigatons or billions of tons of CO2 per year. And so there's a lot of optimism in this space right now. And if you've heard, there's a lot of new startups in this space, maybe looking at enhanced rock weathering, engineering microbes, a lot of new techniques that folks are talking about to capture carbon and put it back in the ground. But I want to also emphasize there are some real naysayers. There's some folks who think that, nope, this is a bubble, that we've really overinflated our ability to restore carbon. And if we could, we would have done it already. And there are some real biophysical limitations. And I'm going to point particularly to this paper by Bill Schlesinger and Ron Amundsen, who's here at UC Berkeley, who really kind of sounded a cautionary tale. So taking that together, it's clear that the jury is out. It's important, it's a critical topic to be studying, but we don't really fully understand whether we can get carbon back into soils. And I would argue that's because we just don't understand why it's there and why it sticks in the first place. So we have a real clear understanding that agricultural systems are sensitive to what's going on with climate change. We hope that they have opportunities for mitigating climate change, but really this is the focus of the work in my group. So let me just walk you through a little bit of a slightly academic backdrop on how we think that persistent soil carbon forms in the first place. And when I say persistent, that's code for long-lasting. You might have heard the word sequester, but I don't use permanent. And I purposefully don't use the word permanent because we know that soil carbon, it's kind of like, molecules in a river, right? They're always moving. They might get stuck in a little eddy by the edge of a stream, but they're really always in flux over geologic time. And we've known this for a long time, actually going back to Darwin and Selman Waksman, who wrote some really seminal work on this nearly 100 years ago. And that's that most soil carbon starts off as dead roots, the parts of plants that get put in the ground. That's where it starts. And those are likely to be retained. The microorganisms actually colonize those roots in an area of the soil that we call the rhizosphere. And they build up all these great kind of sticky, gooey surfaces with extracellular polymeric substances. You have this sort of bloom of organisms that all then die. So that's a whole pile of stuff. It's lipids and polysaccharides, and we call that necromass. So all of that necromass together, it's really kind of sticky material. It does a great job, we think, in actually packing together little particles of soil into aggregates. And those are just little packages that might hold a little bit of organic carbon in the middle. Think of it as sort of a jelly donut. And if you do get an aggregate like that, that might help that carbon that's in the middle get stabilized because it's physically being separated from the organisms that would like to degrade it. And that would cause it to be lost back to the atmosphere. So what causes carbon to persist? Well, I'll tell you, what I was taught in graduate school is it's all about the molecular structure of carbon. It's about whether there's an aromatic ring, whether it's made of lignin, and those kinds of compounds are just really tough for microbes to break down. Their enzymes have to have special attributes. But what we've learned actually in the last 15 years is, nope, it really has very little to do with what particular molecular compound, or we think it doesn't. It has more to do with time and place, whether the right microorganism is right next to the right kind of compound, that it has the enzymatic capabilities or it has the traits to be able to break down. So we're really in this kind of reassessment phase in our field in understanding why carbon gets stuck for any kind of long term in the soil. And so we're doing a lot of experimental work. We're doing a lot of modeling work to try and quickly develop a new paradigm. So I would argue, though, that the paradigm is going to involve these three elements. That we're going to have roots that are introducing carbon, microbes that are transforming it, building their own bodies, and then their necromass also is contributing to forming soil carbon. And then minerals are there. Those are the particles of old rocks, essentially, but very, very fine particles of sand and silt and clay, which are literally kind of the bucket that receives that organic matter. They sorb things onto their surfaces. What you can see in that lovely little SEM images there on the right-hand side is some actual little sausage-shaped microbes that are stuck to the surface of that mineral. It's, I think, a piece of goethite. And so you can kind of imagine, well, you get enough of that going on, that's how you tend to get persistent organic matter forming. So I'm going to kind of talk through some of our work in the framework of these three elements. So first of all, I want to talk to you about deep roots. This is probably one of the most exciting areas for me in terms of trying to accentuate putting carbon back in the ground. So if you look at that image over there on the left-hand side, Panicum virgatum, that's switchgrass. Switchgrass is a native grass. It was growing all throughout the U.S., central U.S. in the prairies that had very, very deep, rich mollisols. And that's where we currently now grow corn. And I should have a picture of corn right there, but the corn roots only go down about maybe 20 centimeters, whereas these Panicum virgatum roots can go down meters, many meters. And they really, if you can imagine, are stuffing carbon in the ground throughout that whole volume. And through a couple of different meta-analyses and modeling studies that we've recently been involved in, we've shown that yes, grasslands, those native prairie type systems, whether they occur in the US or the steppes of Russia or elsewhere, they have a significantly higher both residence time meaning the soil carbon lasts longer, but maximizing the root biomass in those soils is really the most straightforward way to increase soil carbon stocks. And we actually even see that as we project forward in our models of the future Earth, where we have higher temperatures and higher CO2 concentrations. And that higher CO2 in the atmosphere is actually going to be kind of like a fertilizer effect on the plants on our planet, because that feeds the photosynthetic activity. And in grasslands, when you get higher CO2, they actually are just going to pump more of their carbon below ground. So that actually increases what we call the soil sink, or the soil carbon pool. So that's what that little figure there is meant to represent. So conversely, in forests, go ahead.
Oh, so this is not necessarily the long-term persistence. It sounds like there's just a lot of root in the system at any given point in time. Even if it gets eaten, it's still there.
Exactly. And that, yeah. I'm so glad you pointed that out, because it's a really important point that I would say over the last 2 to 3 years, I've come to realize maybe our kind of focus on persistence and sequestration, maybe those are really the wrong words, the wrong notion. It's really not about creating carbon that then becomes old. It's simply continuously pumping carbon into the pool. It's like you've literally got a swimming pool. You don't want it to just sit because evaporation is going to continue to happen over time. You want to have a hose that's always feeding water into that pool and refreshing the stock. And it actually may be quite young, the carbon that's in that stock below ground, but as long as you have roots that are continuously growing and growing and growing over time, that's what seems to maintain that longevity of the soil carbon pool. And again, I want to sort of emphasize that's so important in our grasslands, and that by contrast in forests, a lot of carbon biomass is held above ground. That's wonderful. You've probably heard a lot of hype about planting trees, the Trillion Tree Initiative. You've probably, if you live here in California, you know what can happen to trees, especially if it's dry. We can lose them in an instant if we have a forest fire. So I think of the soil carbon as being somewhat safer place to put carbon. I'm obviously all in favor of replanting trees as well, but there's a trade-off there, as you can see. So I should pause.
Trees.
Go ahead, Greg.
So trees are, what about tree roots? I mean, I get the annual crops have really shallow roots, but any kind of perennial trees, shrubs, and so on, how do they compare to perennial grasses?
Well, I will be honest that we have less data on tree roots and their turnover, but our understanding is they tend to last a lot longer. So a tree invests a lot of resources in its root biomass, but then they tend to last for 4 to 5 years. Whereas a grass, even if it's a perennial grass, is regrowing those roots a couple times a year, actually. There's a lot of turnover and a lot of exudation. So tree roots, the biomass is significant. It's not as significant relative to the above ground bias. The ratio of above ground to below ground is quite different in forests.
Got it.
That makes sense?
Yeah.
Cool. All right. So roots are important. I'm going to give you one example of how they really are important, and the difference between annuals. And if you're not a gardener, let me make sure you know what that word refers to. Those are plants that we replant every year: corn, soy, wheat. Most of the stuff that you're going to go get at Home Depot and put in your garden, you're going to have to replant it every year. By contrast, perennials like that switchgrass, trees, anything that lasts from year on year, those are going to put roots in the ground and they're going to stay there. And sorry, so let me explain the picture that you're looking at here. This is in southern Oklahoma. So land that would have been significantly impacted by the Dust Bowl and lost a lot of its rich organic horizon at the surface. And the plot on the left has been in annuals for decades. The plot on the right has been planted with switchgrass for the past 10 years. Okay, so you can see that, you know, the guy standing there, he's got grasses coming up to his thigh. By the end of the growing season, they'd probably be up to his chest. They get to be about 1 meter tall. So we compared the total amount of soil carbon in those annuals, those are the red dots in the plot there, going down from 0 surface to 3 meters deep. And we compare that to the blue data, which is from the switchgrass plot. So literally just a few feet to the right. And you can see that on the whole, the switchgrass points are have significantly more carbon. We also looked at what we call radiocarbon, the age of that carbon. So radiocarbon is how we do carbon dating. And if you look at that plot, you can also see the blue data are closer to 0, meaning they're closer to modern radiocarbon. It means the carbon that's in that stock has been more recently fixed, photosynthesized, and incorporated into the soil. So that's suggesting that in those 10 years that the switchgrass is growing, it's really making an impact, not just at the surface, but all the way down. Think about how deep, 3 meters, that's 9 feet. That's really a large volume of soil. And when we try to dig in on, okay, why, what could be causing that effect? There are many reasons. One I mentioned to Greg a second ago is just simply the fact that those perennial switchgrass roots are constantly pumping carbon in the ground. But interestingly enough, they're also creating what we call EPS, or extracellular polysaccharides, polymeric substances. These are there are things that roots and microbes do to kind of lubricate their environment, to protect themselves from drought as well. And if you want to think about it this way, it's kind of a sticky goo. It's a sugary, sticky substance, like a mucilage. And when we see more of these EPS compounds, which we do in the switchgrass, we tend to see more aggregation or more packaging, like I talked about earlier, forming those little jelly donuts of carbon and minerals. And then we start to see that carbon lasting longer. So we think that might be one of the reasons that these deep roots are actually causing the carbon to increase. So this was pretty exciting results. We decided to go all over the country and look at these kind of sites where we had old switchgrass stands right next to an annual crop. And so we did literally kind of a road trip. We cored down to 1 meter or 2 in 12 different locations. You can see the little blue triangles there all over the country. And we essentially asked, is that pattern holding? Are we seeing more carbon in soils where we've got these deep-rooted perennials? And the answer is mostly. In not every case, but in the majority of cases, when we look at the switchgrass plot versus the next door neighbor annual plot, we're seeing more carbon, and we're seeing younger carbon using radiocarbon. So all of the places where we're actually gaining soil organic carbon in this plot here are in that kind of shaded area in the upper right. But you can see there's some data plots that fall out of that zone. And interestingly enough, we're trying to understand what causes a site to either gain or not gain. And we think it has to do with how highly weathered, or what the soil types are, what the minerals look like in a particular site. So soils that are more weathered, that have, over geologic time, have actually transferred some of their plagioclase minerals and some of the fresh minerals that you get when rocks are first formed. When they actually transfer those and they create secondary minerals, clays and iron oxides, those kind of minerals are actually kind of more grabby. They're better at holding on to organic matter. So we think that's why some of the sites, particularly in the southern US, are actually literally better at holding carbon, and they have more capacity to gain additional carbon. So we got to be careful though with this kind of data. we did that analysis, and we actually even haven't published it yet, because along the way we realized there was some statistical effects that were making it hard for us to really see clear patterns. And so this is some work that was done by a postdoc in my group, Eric Schlesserov. He's now a professor at Yale. And what he found is there's a mathematical concept that we call regression to the mean. It causes bias in data where you're looking at a time series. And essentially what happens here is that you can make an initial measurement. It might be randomly low, randomly high. It's more likely to be low if we're looking at soil carbon, particularly in a marginal soil. And then you come back, let's say 10 years later, 5 years later, and you measure the soil carbon again. It's possible for that measurement to be just randomly slightly higher. And when you actually compare that data, then you're more likely to be apparently seeing an increase in soil carbon. But really, some of that effect is based on simply statistics that create this sort of exaggerated impression of the soil carbon increasing. So anyway, this is a little bit of a side road here, but just to show you that there's, when you're dealing with big data sets like these, you got to be careful with some of these sort of mathematical effects and how we treat the data. And I would, I put this in because I felt like maybe your group is one that's thinking about big data and both how we can harness that data, but how we also need to be careful in using it.
So actually, it's a great time for a question. So is this a matter of that the initial SOC is a poor or mediocre predictor of SOC 10 years later, or is there a statistical, like, persistent bias?
We think that there's, in any kind of data where you're looking at a time series between one time point and the next, there's some true biological change going on, but there's also a real error in your ability to actually make the measurement. So when we go out and measure soil carbon, we're typically making that measurement on like maybe a gram of soil. And we'll try and make 3 to 5 measurements over an acre, over a field. But it actually turns out that we should be measuring it 20, 30 times to truly characterize that field, because there's just so much heterogeneity on the landscape. So anytime we make a measurement, number one, there's a real variance, and we're not capturing the total population size. And then if we're looking at an effect over time, you can imagine that that's being multiplied. But the likelihood of the statistics are that simply we have more likelihood in the initial measurement to make an arbitrarily low or a randomly low measurement, and then when we come back later, a slightly higher measurement. And so we need to distinguish that sort of mathematical difference from the true biological effect that's going on.
Wait, I didn't follow that last one. Why is it more common to do initially low then high, as opposed to initially high then low?
I think that that's because there is actually the biological effect that is causing a certain amount of carbon to increase. But there's variance associated with both the beginning measurement and the end measurement. And this regression to the mean bias is essentially suggesting that we have, sorry. I'm not explaining that part very well, that there's a random error that makes it look like we're actually seeing a change over time, when really it could be just staying relatively similar, but we have more likelihood of measuring a low value early and a higher value later. I think that I'm not doing a great job explaining this. Let's maybe come back to this at the end, Greg.
Yeah, we should, because
refer you to the paper. I am not a mathematician, but it was a really fascinating effect, and we've seen it in the statistics literature, really well described. And so we tried to bring it into an analysis of our data.
No, that makes sense. And this challenge of measuring soil carbon is very common one. It's very difficult, expensive to collect enough samples.
Exactly, yeah. And I'm glad you kind of are aware of that already. It's funny, 5 years ago, I used to make a joke that measuring soil carbon was the cheapest measurement I make. It's still true. It costs me $2 to $3 a sample.
Right.
But it's not cheap for everybody. And I've stopped making the joke because for landowners and for the carbon market writ large, we've really realized this is a measurement that we need to be making far more than we do right now, and we need to have better models for how to treat the data. We're not in a good place for that. So actually, as part of our Earthshot, one of the efforts we're making is to use infrared or IR spectroscopy data and try and really pull out not just soil carbon, but all of the soil chemistry data that spectra shows us, and use machine learning to try and be predictive to understand, okay, when we have these suite of soil chemistry variables, what is a logical soil carbon? What's the possible soil carbon that we might see in that context? Because there are kind of bounds, upper and lower bounds of how high a soil carbon can be given a particular chemical and mineralogical context.
Right.
Okay, that's a bit of a tangent. Let me get back here.
Yeah.
And it's absolutely fine if I don't get through all the slides. I think it's better that we have a conversation than just drive forward. But I also wanted to talk about another sort of persistent sort of misconception that's out there that we need to push back on in terms of our ability to increase soil carbon. We don't just have an unlimited ability to stuff more carbon in the ground, not only because the minerals just can't hold it, but simply plants photosynthesize at a fixed rate, and we can only grow so much of the time. And one thing we can do, and we show this in our roads to removal analysis, is simply trying to keep plants on the land for more of the year, meaning let's put some cover crops on anytime we're not actually growing a primary cash crop. And that creates photosynthesis and that carbon pumping down into the ground for more of the time. But even given that, there's this wonderful paper that Henry Jansen et al. Published a few years ago, showing that photosynthesis is really the fundamental limit on how much we can increase carbon in our particularly agricultural lands. And they're suggesting that this really is the cap on the maximum sequestration rates. That said, that's one of the things that in our really kind of, trying to be visionary and pie in the sky projects, we have some work funded by the Chan Zuckerberg Initiative, where we're actually trying to change that limit. So with colleagues here at the Innovative Genomics Institute, we're trying to use CRISPR-based gene editing to actually amp up the efficiency of photosynthesis, increasing it by about 15 to 20%. And that would mean not only are we getting a better fixation rate above ground and maybe higher grain production or whatever it is your crop plant is producing for you, biomass, but it's pumping some of that carbon below ground. And so we're working on that right now. We've got it happening in greenhouse-based plants and in gene-edited lines that we've got growing in the lab. And I hope that in another few years, we'll actually be able to transfer these plants out into the field. We're also trying to increase some of their production of those compounds below ground, those EPS production compounds, as well as the length of those roots. So all of that together is kind of where we're going in terms of trying to not just use something basic like cover cropping, but really advanced plants that are able to stuff more carbon in the ground. So the next thing I wanted to talk to you about is, let's say we do that. Let's say we create these wonderful sort of magic plants that are able to put more carbon in the ground. They have these great attributes. How do we actually prove that that carbon is sticking, that it's staying in place? And that's where stable isotopes are important. And that's kind of maybe the through line of my career, to be honest, is using isotopes as tracers and as quantitative tracers of the fluxes in the environment. So we've built a facility here in Berkeley where we can put a plant in a chamber and fill that chamber with the heavy isotope of carbon, so carbon dioxide, or 13 CO2. The plant doesn't recognize any difference at all. It fixes that carbon dioxide into photosynthate, it puts it in the ground, and the microbes that are growing near those roots gobble it up just like they normally would. And that 13C we can track into those microbes, into the soil. We see how much is then respired back to the environment. We can actually even do this in the field. So the picture you see at the bottom there is a site up in Hopland, California, where we did it in a grassland. So I'll give you one example of a study where we did this. We took a switchgrass plant, or a series of switchgrass plants, and put them in these big, tall pots, grew them in a 13C atmosphere, and we subjected them to all sorts of environmental deficiencies, either more or less nitrogen or phosphorus, more or less water, and looked to see where did all that 13C go. And we looked at it both in the microorganisms below ground, but also the metabolites. And metabolites, what I mean by that is really just the compounds the roots are producing. They make a lot of exudate compounds. And I think of these as kind of a chemical call and response conversation that's going on between the plant roots and their microbes. So here's some data that we actually just published last week in PNAS, where we look at really, really complex suites of metabolites. This is measured with LC mass spectrometry, and then also really complex suites of microorganisms, and tried to see data-wise, what's the correlation? And here we see really a beautiful correlation that's based on the environmental constraints that we put in our different treatments. So what you see at the top there that I've highlighted in yellow are all of the soils where we added nitrogen, or we added nitrogen plus phosphorus. We're getting not only a different kind of suite of organisms that are popping up, but very different metabolites being produced. As compared to the control soils, which are outlined in blue there, again, just different microorganisms, different metabolites. And this is a multivariate regression analysis. I will admit right off the bat that regression is not, it's not entirely satisfying as a mathematical tool to correlate these two extremely diverse data sets. But it's kind of the best we have right now. And again, I just put that out there as a challenge to your group, because you're probably way ahead of me in terms of thinking about these kinds of things. But we really are desperately in need for finding new ways to take two very, very complex data sets, or even many data sets, and look at inference of interactions between them. We use network analysis and things like that right now, but I would say most of us in my field right now feel very unsatisfied in terms of our ability to predict metabolites with microbes, or predict microbes with metabolites. We'd literally love to be able to do that better. Still, that said, we're showing here that there's really an effect on the soil microbiome that's driven by those edaphic conditions, whether there's nitrogen available or water, et cetera. So to really harness plant traits and harness the kind of crop plants that we're producing and growing on the land to put more carbon in the ground, I would argue we really need a multi-prong effect. We need to both increase the amount of carbon simply that's fixed, maybe in photosynthetic efficiency, increase the carbon flow into the ground. That might be more exudates that's stimulating those microorganisms, more of the sticky goo compounds that are binding soil particles together. And we need to think about how to actually then retaining that carbon. I mentioned earlier, we largely are interested in simply increasing the flow, the persistent pumping of carbon below ground. But I think there are ways that we can amp up how much of that carbon gets really held on in a longer term sense. So I'm going to transition to talking a little bit more about those attributes below ground in the next part of my talk. But let me pause and just ask Greg, if you have any more questions on this part.
No, it's good. I'm looking forward to that part.
Okay, all right. So let's talk a little bit about microbes. That's I'm trained as a soil microbiologist. That's definitely where I've come from. And again, I want to reemphasize that 50% or so of the soil carbon is actually made of microbial bodies. It's what we call necromass. There are those little sausages on the left there. Those are probably some bacilli, bacteria. On the right side here, you're actually seeing the leftovers of fungal cell walls. So this long little thread there that you see is a fungal hyphae. And if you have a compost pile, I bet you've seen these. They're really fine little white threads that you probably have seen growing in your compost bin. And these mycorrhizal hyphae, there's an enormous amount of them in soil. They stick to mineral particles. They create a lot of also polysaccharides that glom things together. And we think they're really important as essentially kind of highways for transporting other microorganisms and water throughout the soil. And obviously once they're dead, their biomass or their necromass persists. So we have a whole list of different traits of microorganisms that we've speculated might be important in predicting soil carbon. And no worries about reading through all of these different compounds, or sorry, all of these different traits. But we came up with this list largely based on the fact that in the last decade or so, we can now measure all of these traits simply using genomes and transcriptomes, the expression of genes in microorganisms. And we've had really just a revolution in our ability to sequence the whole metagenome of a soil. So extract all of the DNA and try to repackage together the individual genomes of the organisms that lived in that soil. We can usually do that for several hundred to maybe 1,000 microorganisms, and then read that library, as it were, for all of the different functional traits that those organisms have. And if we could really not just read it, but understand what those genomes were telling us in terms of the functional potential of the soil, we really think we would be in a much better place to be able to predict what's going on with soil carbon. Probably lots of other biochemical processes as well. So we really focused on several of these traits in combination with our isotope tracing. And let me just say another word about why we do isotope tracing of microorganisms. At any given point in time in soil, we are only able to culture maybe 1% of the soil microbiome. We're just honestly terrible at it. And folks have tried and tried. Maybe we can get that up to 3 to 5%. But the vast majority of microorganisms, we can't actually get into the lab and grow by themselves. They want to grow in situ. They want to grow with each other. The other thing that we found when we use our isotope tracing, whether it's adding 13C with a plant or just adding 18O water, so heavy water that any living and dividing microorganism is going to take up into its DNA, we find that often only like 8, 20, maybe 30% of the total microbiome is actually active, meaning only that small subset is dividing, responding to the environment, and actually growing. A huge amount of the soil microbiome at any point in time is just dormant, just quiescent. It may be that even some of that DNA is not even within a cell wall. It's just swarmed onto a mineral surface. So it's kind of a relic of previous conditions. But so because of that, because only that small subset is active, that's why we think it's so critical to use what we call stable isotope probing, or these methods where we're actually able to pull out just the pool of organisms that are active. So almost all the rest of the work that I'll tell you about, we've used that particular technique to really subset the active organisms. And in a study we did recently, we were looking at how that active microbiome was responding to either living roots growing into a soil or roots that were starting to decay. And this is work led by Noah Sokol, who was a postdoc with me at the time. And it really, he theorized that it matters whether you've got microbes that are actually growing on the surface of organic matter, and their traits are causing that new organic matter produced by roots to be turning over really quickly. So those microbes might have really fast growth or high carbon use efficiency, versus different traits occurring depending on whether they're living near a living root or a decaying root, or even out in a bulk soil where there's no root at all. And he theorized that, yeah, maybe it really matters where you live if you're a microorganism, that you might have very different traits depending on whether you're in that sort of fast-paced zone where there's lots of roots being pushed out by excuse me, lots of exudates being pushed out by roots versus out in a bulk soil or near a decaying root. And what Noah showed with some isotope tracing is absolutely it does, and that many of the traits that we can both measure directly in the soil, but also measure in the genomes of these organisms are different depending on whether you're near a living root, that's the data on the left-hand side in the green, versus near a decaying root. And that the amount of extracellular polymeric substances is substantially higher near a living root. Growth rate is also higher. The amount of simply microbial biomass tends to be higher. But interestingly, the carbon use efficiency, how much carbon an organism is keeping versus letting go, can be substantially lower. So that's really starting to tell us that these traits that we can measure in these genomes of these organisms, particularly the ones that are growing, can be really different depending on where the habitat is of a soil. That's not how we model this right now. Currently, we tend to model soil microorganisms as if they're evenly distributed across the volume of the soil, and that we're showing it's really not true, both in space or in time. And the other thing that I'll just point out, the data that's there on the bottom right, is that actually only about 50%, a small proportion of all of the organisms that were found in the soil, I want to say it was about 5 different phyla, were responsible for over 50% of the carbon assimilation in the soil. And the reason that matters to us is that when you think about soil microbiomes, they're incredibly diverse, just thousands, tens of thousands of different phylotypes, different species, as it were. But what we're showing is actually there's a really quite limited number of organisms that are active at any one point in time. So we don't necessarily need to understand everybody to be able to predict what's going on with the whole soil. We really just need to, again, focus in on that active portion. So when we try and really scale this up to understanding what's going on with soil carbon, it's super critical that we stop just have the blinders on and thinking only about bacteria, only thinking about fungi, only thinking about viruses. That's kind of been the traditional way that microbiology has approached environmental work. That's how we tend to be trained. But work started some years ago now by Evan Starr, and that has continued in our group and others. We use that isotope tracing technique to pull out all of the genomes that are enriched. So not just bacteria, but also the eukaryotes, meaning the protists, the other kinds of nematodes, and even viruses. So phage are proving really, really important in the turnover of soil carbon. And again, we're still kind of in early days with the bioinformatics, but being able to predict what viruses are targeting what hosts, whether they're bacteria or fungi or protists, is proving really important, because those viruses, just as they do for humans, are infecting their hosts and causing a lot of damage, causing a lot of lysis, a lot of loss of carbon, a lot of death. And that turnover is incredibly important for the cycling of carbon in the soil. We've literally learned that in the last year or two. But we're still early days in being able to predict which virus targets which host. Right now, we're using that CRISPR sequence, which is kind of the immunity system for bacteria, but that we unfortunately only find CRISPR sequences in about 20% of our genomes. So we really are needing to find new ways to be able to do that kind of prediction. But that said, I just wanted to make a point for let's focus more and more in the future on sort of whole cross-kingdom interactions and not just one segment of the microbiome. Okay, let me pause there, see if there's any questions, Greg, before I keep going.
Well, one thing is coming up, you're going through this the diversity of organisms, but you've not talked about fungi, which seemed like another very interesting mess that it complicates all these dynamics. And in fact, that's your next slide.
Yeah. Okay. Good. Well, that's great segue. That's exactly what I was going to talk about next. Just a couple slides here. I know we're getting further along here in the time, but I wanted to say that probably the most exciting space that I'm interested in, if I was going to focus on one type of microorganism and its role and its traits in predicting or promoting the ability to increase soil carbon, that would be mycorrhizal fungi. So mycorrhizal fungi are some of our oldest associates of land plants, particularly our arbuscular mycorrhizal fungi. They infect 90% of the species on Earth. And they provide a huge amount of nitrogen, phosphorus, even water to their host plant. In return, the plant shares a little bit of its carbon, its photosynthate, its sugars with the fungus. And that allows the fungus to not have to degrade organic matter. It can kind of feed off of the plant, and they have this symbiosis. Now, these hyphae grow out from the roots. If you've ever seen it, there's these wonderful photos of what a single root might just be fairly limited in space, but it could have thousands of actually centimeters of hyphae extending from it. And we actually did an analysis recently where we calculated what that total length might be. And for fine roots, it's something on the order of 7 centimeters of root per cubic centimeter of soil. But the hyphae that are, again, symbiotic with those roots and extending even further, 102,000 centimeters per cubic centimeter. So almost 2 orders of magnitude more of total length of exploration that's going on because of these fungi. And in another isotope tracing experiment that we did recently, we really showed how important that length of hyphae is for transporting carbon. So first of all, we set up this kind of wonderful design here. This is Anne Kakaridis' thesis work. And she had 2 pots with little air gaps so that only the hyphae can jump over the air gap. You got a plant on one side, no plant on the other. And special mesh that only allows those little fungal hyphae to creep across the air gap. And if you look at the bottom right of your screen, you can kind of see that kind of, sorry, not great resolution image, but those little threads there are fungal hyphae. And that was taken with my iPhone, looking through a microscope viewpoint. And you can see that those hyphae are really kind of sticking together and holding together lots of mineral particles. And we then did some other kinds of isotope imaging of those hyphae and other measurements, and we found those hyphae are transporting a really significant amount of carbon out into the soil. So over just 6 weeks, they increased the amount of carbon in the non-plant side by 2%. That's a really short period of time to have increased the carbon that much. Then we took some of those hyphae that were over there, and we actually did our stable isotope probing technique. We were able to reconstruct whole genomes of multiple organisms, and we were able to show there's this whole wonderful kind of food web going on surrounding those fine little threads of hyphae. We've got different organisms that are predators, that are preying on bacteria, that are probably preying on the hyphal material. We've got ammonia oxidizers. We've got a whole milieu of different microorganisms that are being stimulated just by those fungal hyphae growing out into the soil. And so that's a really exciting kind of new understanding for us of how complex the system is surrounding hyphae. But for me, it's also suggesting that these mycorrhizae are important ways of pushing carbon further into the soil than just roots do. And I think that again, is something that if I were to focus on really adapting agricultural systems, it would be really trying to amp up the efficiency of mycorrhizal colonization.
So then pushing on this, so I see the transport potential. And now what about themselves storing carbon? Because I know they don't respire the same as plants, but they're built of carbon. So is that an opportunity?
It is, and we've actually known for some time now that the cell walls of those mycorrhizal fungi are really rich in compounds. Chitin or N-acetylglucosamine is one of them. It's a kind of amino sugar, and that those compounds seem to stick really well on particularly iron oxide surfaces. They seem to really last. Again, it's not because they're impossible to break down, but they seem to be just particularly good at bonding to the mineral surfaces. And if that's really true, and we accelerated the production of that kind of biomass, not only would it probably help the plants, because the more mycorrhizal colonization, the better the plant nutrition is working, but it might also end up pushing more carbon into those persistent soil carbon pools.
So
Excuse me.
then, I mean, the obvious extension then is you're trying to push plants to perspire more carbon. It seems like fungi may even be more flexible.
They are, and there are lots of things that that I think we can harness in the fungal world, but we need to be a little bit careful because the fungi, they are symbionts in that they're benefiting the plant, but there are many of these fungi that actually also have the ability to kind of turn around and decompose the plant. So it's a little bit of a Jekyll and Hyde relationship. And I think we need to better understand when are fungi reliably on the sort of benefit the plant, benefit soil carbon side of the ledger versus what causes fungi to turn around and be like, oh no, I'm going to break down my host because that's free carbon. Yeah.
Thank you.
So Greg, tell me, I've got another few slides that I could go through, or I could just kind of stop or skip to the end and just answer questions. It's your call.
Well, I guess what's the topic?
Well, the rest of it is kind of focused on the minerals and the modeling that we've done, kind of getting back to the national scale on the ability of certain soils to be really the ones we should focus on increasing soil carbon.
Well, that would be awesome, because actually, my next question here is also about them and biochar, because that's the other thing.
Biochar. I didn't put any biochar in here, but okay. Let me skip ahead to the slide that's. Okay, let me start here. Well, okay, I'm gonna start here. So I want to emphasize that we don't have just an infinite ability to grow the soil carbon pool. We can't just increase. Yes, there are soils that are almost 100% soil carbon. If you go up to the Arctic, or if you're in a wetland, you can get 70 or 80, even 90% soil organic matter. That's because you have anoxic conditions or it's very, very cold. But in most of the places that we're practicing agriculture, and we therefore have the ability to really change our practices to increase soil carbon, it's a mineralogical aerobic context. And if you look at that data on the left-hand side, Francesca Confruccio has showed really nicely there. This is global data, but it seems to hit kind of a threshold of the amount of carbon that can be associated with minerals. So we think of that as being a sort of a saturation point. There are, however, differences in the rate of accrual, meaning how much new carbon we can capture. So if you think about those soils from the Dust Bowl that I showed you earlier in Oklahoma, where we were able to increase soil carbon by planting switchgrass, those soils are accruing new carbon largely because they're not at their saturation point. They're somewhere much, much lower. They lost most of their organic rich horizon. So what we would love to be able to do is really predict, where are those sweet spots in the United States? Where are the places where we have the ability to gain a lot of carbon and relatively quickly? So that's what the point of this paper, again, Eric Slessor's work is to really try and predict, where do we have the great sort of mineralogical capacity to capture carbon. And Eric focused on where we have soils with these short range order minerals. They're minerals that are really good and grabby at holding onto carbon. And you can see that both in the Pacific Northwest, as well as a lot of the eastern half of the Midwest, has really excellent capacity to grab onto soil carbon. And so the big modeling effort in my group right now, as part of our new Energy Earthshot, is really trying to get down to even 10-kilometer or possibly even better scale, and predict whether a region has the right climate to grow and have high productivity of plants, but secondarily have really the right kind of chemistry and mineralogy in the soil in terms of its cations and its pH to really hang on to the carbon that's produced below ground. Because that would allow us to get to a point where we have a policy scenario where, you know, maybe particular counties or particular states would be really incentivized, because those are good places to really focus on soil carbon.
And just to be clear, this is now enhanced rock weathering.
This is not enhanced rock weathering. This is simply the inherent mineralogical capacity that already exists within a site. Yeah. Yeah.
Okay. So this is like rocks that are already there, ready to receive the carbon.
Yep. Yep. But I'm gonna let me just put, because funny, you're great, Greg. And I'm guessing what my next slide is. I'll just maybe kind of show this one and then end up, because I know we're near the end of the hour. Enhanced rock weathering would be kind of icing on the cake. That's literally trying to goose the ability of soils to capture carbon. And the idea here is we use crushed basalts, these fairly reactive rocks that are really great at interacting with CO2 in the atmosphere, and the magnesium forms bicarbonate ions and potentially even carbonate minerals that pull CO2 from the atmosphere down into the soil. Now, it really depends what the pH of the soil is, whether you actually get literally formation of new rock in the soil, carbonates, versus just bicarbonate that's going to probably end up washing out through the soil and into the oceans. But regardless of which of those pathways happens, we're pulling CO2 from the air down into the soil system. Those are all inorganic reactions. We might also have organic reactions that are going on at the same time. That's one of the things that we're studying. We really just don't know. And it could be some of the acids or exudates from roots or microbes actually kind of goose that process and make it occur even faster. So again, that's kind of a whole suite of, excuse me, a whole suite of questions that we're asking in our new Terraforming Soils Earthshot project. That work is really just getting going right now. We're starting our field trials next month. I'm gonna skip to my end slides here. These are some opportunities. I'm happy to share these slides with you, Greg, just so you know.
Awesome.
Opportunities that I see in their near future. They range from those mycorrhizae, focusing on photosynthesis and EPS. But I also wanted to just end by talking very, very briefly about what we showed in Roads to Removal, which is all of that great stuff that I just told you about is in the research phase. It's going to take us 5-10 years to get it to a point where we're actually applying it in the field. What we showed in our Roads to Removal analysis, though, is that there are things we can do right now. And the cover cropping is the key one. Planting those perennial crops is also important. But I want to make sure that we don't get so excited about what's coming in a decade that we forget to do what we know how to do right now. And because it's so critical that we start removing CO2 literally today, we really want to emphasize that some of these known practices, like putting crops on the ground in the wintertime, is a really critical thing to do while we kind of bridge towards some of these really novel techniques that the Earthshots are supporting.
All right. Well, yeah, thank you very much.